Hi, I'm Diogo, member of the Sensory Ecology Lab in UFRN Brazil, and today I'm going to present my work entitled The Role of Carapace Coloration in an American Fiddler Crab. Fiddler crabs live in a transition environment such as mangroves and beaches. During low tides, they leave their burrows in the sand to compete for resource, eating and find mates. All this occurs in a mixed population with hundreds of males and females at the same time. In this context, it's important to recognize conspecifics and avoid heterospecifics, as the males invest a lot of time to attract females. They attract females waving their big claws in a reproductive display, called waving. In the northeast of Brazil, there is a species called Leptuca leptodactyla. Here we can see the yellow of its claw and the white of its carapace. Now look at this heterospecific male. This is another species of the same genus, Leptuca cumulanta. Its claw is gray and its carapace is dark green. These two species are similar size but different colors, and their populations partially overlap. So how do females of Leptuca leptodactyla recognize its conspecifics? Is the carapace color used by fiddler crabs to identify its species? This study aimed to comprehend how carapace coloration of fiddler crabs from America affects interspecific recognition. We hypothesized that carapace coloration of males may give a signal to females, directing the choice to conspecific males. Now, how did we test female preference? By giving Leptuca leptodactyla females a binomial choice of conspecifics and a heterospecific males with claws and carapace reflecting different colors. To perform that, we built an experimental arena with a lever mechanism. With this mechanism, we are able to release the females from a distance, and with that, female can either walk towards a male or just leave the arena. Let's take a look at the real experiment. Here we can see the female leaving the arena . And another female choosing a male. We consider a choice when a female approached within at least 2 centimeters of a male. Each female was tested only once, and males were used until 3 choices of different females were made. After each choice, we switched male positions. The male claws are always painted yellow, and the carapace was painted white or dark musk green. Since male claws were always painted yellow, female can only use the carapace colors to identify conspecifics. After we repeated this experiment several times, look what we found. This graph shows all results, where each panel represents our experiments. Let's look at the first one. It shows female choice by the male type. This is the conspecific with natural white carapace, and this is the heterospecific with natural dark green carapace. The female significant preferred the conspecifics. Both claws were painted yellow, so females were only use the carapace color to make the choice. Now, let's take a look at the second experiment. Here we have the same species, but now we painted conspecific males white and heterospecific males dark green. Notice that this is their own colors. In this experiment, the female continued to prefer the conspecifics, which is expected once we just painted them to similar to themselves. Finally, let's take a look what's happened when we change their colors. Again, here we have the same species, but now we painted conspecific by heterospecific colors and vice versa. So the conspecific Leptuca leptodactyla was painted dark green, and heterospecific Leptuca cumulanta was painted white. With this change, the females start to prefer the heterospecific male instead the conspecifics. That's very interesting. What shows us that carapace colors can be a signal to conspecifics in these species. And they can recognize conspecific only by carapace colors, which makes sense once males sometimes lose their claw competing or dealing with predators. We know that interspecific recognition is crucial for the reproductive success of most animals, as it avoids wasting time and energy in new interactions with individuals of different species. We also know that some fiddler crabs are capable of perceiving colors and use coloration for interspecific communication. Until now, most studies have focused on the claw coloration, and the usual function attributed to Carapace coloration was camouflage against avian predators. Here we show that female can perceive signals coming from male carapace. That collaborators in 2007 showed that male can differentiate female neighbors from unfamiliar female based on carapace coloration. Here, we add to these results that female can use carapace coloration to identify conspecifics. Received signals from carapace in Leptoleptodactyla can go beyond interspecific recognition and help distinguish between conspecific males. In Leptoleptodactyla, males in reproductive period change colors to a bright white and spend a lot of time attracting females to its burrow. In conclusion, this is the first study that demonstrates that females can recognize the conspecific through carapace coloration. Thank you for your attention. I'm available on this email address in case there are any questions.